how crucial biochemical reactions are in our body. So, and we also categorized that the whole of biochemical, this whole set of biochemical reactions that happens in our body happens as metabolism. And metabolism, we divide it again in two forms. One is catabolic process, other one is anabolic process. Correct, right? So we understood that in both the process, what happens is in one process we have is the release of energy, and in the other process we have is the consumption of energy. Correct? Whereas if you look at any chemical reaction that is happening in our body, it requires a tremendous amount of energy, especially for breaking, especially for let's say for construction of something. If you want to make proteins, if you want to make polypeptide chains, or if you want to make complex lipids, or if you want to build cholesterol, or any of the nucleotide chains like or nucleic acids, it requires a lot of energy. Similarly, and there are some complex molecules. For example, if you take cellulose, if you take starch, if you take any of the lipids, to make them into simple forms, for example, let's say that we have water, and the water is used by your plants, correct, right? For the process of photosynthesis. Here it goes under photolysis process, correct, no? So if you see in your body, for example, if you have carbon dioxide and carbon dioxide mixes with water and it results in carbonic acid, H2, H3CO4, okay? So carbonic acid. So similarly, any of these chemical reactions that happens in your body, so here it requires a process called as making and breaking the bonds. To make and break the bonds, simple, if you take nitrogen, nitrogen has three bonds, covalent bonds. To break the nitrogen bonds with the energy, physical energy, it requires a lot of pressure and lot of temperature, around 700 degrees Celsius of temperature and very high pressure to break this covalent bond. Can our bodies afford this much investment of energy? That is the first question. So we can't invest. See, the amount of gain we are getting out of what we eat is very, very less. If no enzymes are involved, or if none of the biochemical reactions are helped by something called as enzymes, so then we would be investing almost 100 to 1,000 times more energy, okay, to run the system. You understand? So like your bike has to give you mileage to reach a certain distance, okay? That it has to be efficient. When the system is efficient, you conserve energy. So conservation of energy is very, very important, which we cannot just invest all of this for only biochemical reactions, correct? No, our body has several roles. For example, you think, you repair, and you regenerate, and you divide. So many functions are happening apart from your biochemical reactions. For all those things, you need energy. So how is our body conserving energy? Like that of any chemical reactions that we see in the industrial scale, like the smelting industry or the iron industry, anything you take. Among all this industry, what is happening here? Our scientists or the engineers, they don't go for complete pure process. The pure process in the sense, they don't just go for example, if I have to go for smelting of iron or purification of iron, do they not use catalysts? So we have heard about the application of catalyst for the process of all chemical reactions. Most of the chemical reactions, whether it is in industry or in any laboratory, correct? No? So why do we use catalyst? Catalysts are the substances which fasten the rate of reaction, correct? No? So similarly, our body is like a factory, and a lot of chemical reactions are happening within our body. And one of these systems that reduces the application or use of energy is our enzymes. Okay. So what are enzymes? Enzymes are also called as biocatalysts or biological catalysts. They are natural catalysts or organic catalysts, we can say. So if you observe any living organisms, guys, what we can see is our body's function is 98% controlled by something called as proteins. Okay, proteins are responsible for 98% of all living functions. You might have studied all these biomolecules, but you see the major role, like what? Transportation, recognition, acting like receptors, okay, and antibodies. And if you see so many functions, like you name any of the things, like the what do you tell, the the fibers in your skin, okay, the fibers in your muscle fibers, the contraction of muscles. So if you keep on listing all the functions and all the components in your body, 98% of the functions are carried by proteins. So with this information we can tell that enzymes are proteins. Enzymes are nothing but proteins. They are nothing but specialized proteins. So these specialized proteins, they are very important to fasten up or speed the chemical reactions. Understood? So, how what are enzymes? Enzymes are colloidal in state. So, if you see a colloidal, what is a colloid? For example, if you take a chalk piece, and then make it a powder and mix it in water, how does a water appear? A water does not appear completely like a pure solution. It looks colloidal, like milky, or like curd. If you mix curd and water, so that's what the colloidal state is. So it looks like colloidal in nature. And moreover, it's a protein. What did I tell? Enzyme is nothing but most of the enzymes are protein. What is one important feature of protein? They are structural molecules. Okay, they're highly structural molecules, just like your lock and key. Okay, so the structure and being a protein makes them very specific, very specific, in the sense they only act on the things that they're meant to. Okay, so these are the two things. Next one, if you see the enzymes are not always active enzymes. When they are released by our cells, they are released in the form of proenzymes. Proenzymes means the immature enzymes or inactive enzymes. The proenzymes are also called as zymoenzymes. Clear? For example, you don't have it in the syllabus. If you read the further chapters, like we had digestion in the last year, so I mean a few 2 years back, I think so. Yeah, 1 year back. So we had digestion and absorption. You could easily make out that our pancreas, which produce insulin, correct? No, our pancreas produce what? Insulin. You might be aware of it. What is the other function of pancreas? Pancreas produces so many different enzymes like procarboxypeptidase, and then you have nucleases, proteases. All of these enzymes are produced by your pancreas. When the pancreas produces the enzymes, so the pancreas used to produce in its most inactive state. The enzymes were completely inactive. The reason being, they will go to the site of action. That means they have to go to the place where their role is supposed to happen. There they get mixed up with other components and or they're converted actively. One more example I will give you. You have a stomach, correct? No? The stomach has what? It has HCl, correct? And what does the stomach produce? It produces something called as pepsinogen. Pepsinogen is an enzyme. Okay. Now, this pepsinogen is in its inactive state when it is produced. Now, what happens is when your stomach releases this HCl and its pH reduces, when the stomach pH reduces, this pepsinogen is converted into active pepsin. You see, we have understood how the enzymes are produced in our inactive state and how the inactive enzymes are called as the proenzymes and the proenzymes are converted into active enzymes. But here, as I told you in the beginning, enzymes are proteins. All enzymes are proteins, but not all proteins are enzymes. It's very simple. So if you see enzymes, whatever the enzymes you come across, most of the enzymes will be proteins. But if you see all proteins, are they enzymes? No. If you get confused with the statement, as I told you, in protein can be a fiber, it can be an antibody, correct? No, or it can be your enzyme, it can be your receptor. Okay, receptor. Example I'm telling. See, a protein can be all of these. Okay, what about enzymes? Most of the enzymes, their composition is made up of proteins. Okay, that is what the statement is. Remember this statement. I know that this is a very simple statement, but when you read it fastly in your question papers, there are chances that you might choose a wrong answer. all enzymes are proteins. All known enzymes are proteins, but not all proteins are enzymes. So this or arrangement of statement is very, very important. But there are exceptions. What are these exceptions? There are some enzymes. You remember these names of these enzymes? Ribozyme, okay? And then you have ribonuclease, and then you have peptidyl transferase. So these are the three enzymes which belong to non-protein category. So these, what are these ribozyme? What is ribonuclease? What is peptidyl transferase? So their role, okay, their role and what they do, you will study in the second year, okay? When you study DNA replication or transcription or translation, you will come across this, and I will be teaching you what are the rules. But remember these names: ribozyme, ribonuclease, and peptidyl transferase are the only three enzymes that belong to non-protein enzymes. Rest all known enzymes are proteins. Clear, guys? Okay. So what does enzymes do? So the enzyme is just like an intermediate. For example, if I have glucose, okay, and if I have to convert this glucose into glucose 6 phosphate, or if I have carbon dioxide and I want my carbon dioxide to react with water, for the possibility of my carbon dioxide to react with water directly, it requires a lot of conditions. What conditions it require? It might require a specific temperature. Correct? No, I will write it here. It requires a temperature, or it might require pressure. It might require pH. So many factors will judge how a chemical reaction has to happen. No, so these requirements will be very, very high that our body will not be able to provide it. Got it? So what can enzyme do? If this is the case, there is an enzyme called a CA. Okay. So this, just to remember, let's say that we have an enzyme called as CA. So this particular enzyme, what it will do is it will make sure that it has some surface, that structure. So the structure makes the carbon dioxide very attractive towards it. Got it? No? So our substrate is what? Our substrate is our carbon dioxide. And our enzyme is what? Enzyme is our carbonic anhydrase. That is the enzyme CA. Okay. So now what will happen? Once these two are present, now they convert themselves into substrate enzyme complex. Okay. What is a complex? A complex is a union or combination of your substrate and your enzyme. So why do they make this complex? The enzyme has such a structure that it attracts your substrate. Bonds are created within this enzyme and substrate. So that will help in the reaction of other intermediates. What are those? What is our other thing that has to react? Water. So when water could easily react with enzyme substrate complex than directly reacting with carbon dioxide. You understood? So what happened with enzyme substrate complex? Enzyme substrate complex created a room for faster, easier, less energy, and better chemical reactions compared to direct interaction of the two molecules. Understood? So that will result in what? The carbonic acid, etc., etc. That is, it is not required. Okay. Now, let us take one example of a simple biochemical reaction that happens in most of the cells in your body. So the event that you are seeing here, that is the chain of events that is presented on the slide here, is a process called as glycolysis. Glyco Glycolysis. What is glycolysis? Glycolysis is a process in which our cells, okay, use the glucose, okay, to create ATP molecules. Okay, it has to use glucose to make sure that it will produce what? ATP. Correct? No. Why do we use glucose? Why do our cells use glucose? Our cells use glucose to get energy. What is our currency of energy? ATP. So how does it do it? So now the cell takes glucose. Now, can it directly burn the glucose into, sorry, can it directly burn the glucose and provide us ATP? It's not that easy. You see, a chemical reaction, a biochemical reaction is a stepwise. You see, just taking the glycolysis, it's around 10 to 12 steps are there. A cascade of steps happen one after other, something like that. So how does it take place? See here, if you see the glucose, the formula of glucose is C6H12O6. And if you observe in the beginning, we have 6 carbon atoms. Now, I have told you guys that when you break bonds, you make energy. And our body learned that if I make sure that my glucose is broken down into 3 carbon atom molecules, I convert this C6 into C3 and C3. I'll create 3 molecules. Okay, if I break this carbon, I can create 6 divided by 2 is 3 and 3, no? So I can get 2 molecules of 3 carbon containing compounds. So the whole of glycolysis, what happens? A 6 carbon atom glucose is converted into a 3 carbon atom pyruvate. So conversion of glucose into pyruvate will give us what? So this is our spelling, okay? Pyruvate. So conversion of glucose into pyruvate gives us what? ATP, which is our energy. Now, this is not a simple process. It has to happen. A series of process has to happen for this conversion. Why this so much reactions has to happen? So that my body can conserve energy. That is our main goal. You invest less energy and you gain more energy. When you invest less energy and when you gain more energy, then your industry will be in profit. Correct? No. If I invest some money, I have to make profit, right? Only my industry will run. Similarly, if I invest some energy here, then my net gain should be what? More than what I have invested. That is when I can run my system. Otherwise, I will collapse. If I go under loss, my cells will die. Simple. Correct, right? So that's what is happening here. So you see, the process takes place like this. The glucose is converted into glucose 6-phosphate. The glucose 6-phosphate is converted into fructose 6-phosphate. The fructose into fructose 1,6-bisphosphate. Like this, a series of reactions will be happening. To make sure that these reactions happen with a very minimalistic energy, each of the step, you can see that it is supported by the presence of an enzyme. You can see here, for the conversion of glucose into glucose 6-phosphate, it's a simple reaction. What is glucose into glucose 6-phosphate? You have 6 carbon atoms. Just to make you understand, okay, you have to study glycolysis, but this will give you a foundation. Okay, 1, 2, 3, 4, 5, 6. Okay, guys. So what I'll do, I will mark this carbon atom. 1, 2, 3, 4, 5, 6. I have 6 carbon atoms. Okay. Now what should I do? Glucose 6-phosphate. You see here, glucose is converted into what? Glucose 6-phosphate. So at the sixth position, a phosphate molecule has to be added. It is a simple process. I take a phosphate and I add it to the glucose. For this, to create bond, I have to invest energy, correct, right? So I invest energy, very little energy. And why am I investing very little energy? Because I am supported by an enzyme called as hexokinase. You see, hexokinase. So like similarly, for every single transformation, there are enzymes. You can see here. The next step, from glucose 6-phosphate to fructose 6-phosphate, what do I have? I have phosphoglucose isomerase. Isomerization. You know, glucose and fructose are same formula, but their geometrical structure is different. So isomerization reaction will happen. It will change its, structure, but the same formula. Similarly, you have phosphofructokinase, aldolase, okay, ACE. You can see here all hexokinase, isomerase, phosphofructokinase, aldolase, okay, and pyruvate kinase. Similarly, all these enzymes end with ACE are the supporting molecules to make sure that this glycolysis or any biochemical reactions happen. Clear, right? Are you following me, guys? Hmm? Clear? So that is the role of enzymes. Now we'll understand the role of enzymes even better. So you can see that how they come up at each place. They reduce the requirement of energy and they speed up the rate of reaction as well. Okay. Next one. If you look at the enzymes, as I told you, we just saw. Excuse me. You just saw a few examples. Okay. Where's my ink? Yeah. Yeah. Sorry, guys. Yeah. If you look at our past few enzymes that we just looked into, hexokinase, phosphoglucose isomerase, then you have aldolase, you have fructase, and then you have phosphoglycerate mutase, ACE. Okay. So all of them, anhydrase, isomerase, dehydrogenase. So all of them, they end with the end term called as ACE. Okay. Anything that comes with ASE are called as enzymes. Okay. Next one. One thing we use, we make sure that they end up with the last few letters called as ACE. Correct, right? And the second criteria for naming the enzymes is based on the two major category. Okay, two major criteria. What is that? One, on what substrate it is acting on, and the second one is what kind of chemical reaction it is doing. It's so easy to understand here. You can see here in this particular case, you have something called as isomerase. Why did we call it as isomerase? Because it is converting glucose into fructose. So this process is isomerization reaction. Geometrical isomer is geometrical isomer is changed to fructose geometrical isomer. So this reaction is called as what? Isomerization reaction. So who does the job? Isomerase does the job. So this is a type of chemical reaction. If I remove hydrogen, dehydrogenase. You understand? If I remove carbon dioxide, decarboxylase. Okay. If I do oxygenation reaction, oxidase. If I do reduction, reductase. Okay. If I transfer something from, see, if I transfer this carbon from here to here, it is called as transferase. So these are called as type of chemical reactions. You understand? So in the same way, you can see here, you have phosphofructokinase. Why it is called as phosphofructokinase? Because it is fructose 6 phosphate. It acts on fructose 6 phosphate to give us fructose 1,6 bisphosphate. So what is its action? It is acting on a fructose 6 phosphate. That is why the enzyme name is called as phosphofructase. So for example, if you have an enzyme, it can be named on two important criteria. One is the substrate it acts on. Second one is the chemical reaction it is doing. Examples for the substrate it acts on is maltolase, cellulase, okay? And then you have phosphofructase, etc. If it is a chemical reactions, it will be hydrogenase, decarboxylase, isomerase, reductase, oxidase, etc. Okay, so this is how the nomenclature of enzymes happen. Are you guys understanding? Are you guys following me? Yes, no, guys? Very sleepy? A little while. It's an introductory phase, no? Anyways, are you following me? Yes. Perfect. Thanks for the response. Others, I hope you are following too. So you can just give a thumbs up, a reaction. Okay, I will follow everything that you are trying to communicate me through. Chalo. So the next one is, how do enzymes function? The thing is, enzymes are structural molecules. Why the enzyme? Structural molecules. Because I don't understand what do I mean by the structural molecules. Let me ask you a simple question. Okay, I hope you can hear me well. Let me ask you a simple question. What is your RBC made up of? Okay, what is your RBC? Red blood cells is made up of what? Why is it red in color, guys? Anybody? Why is your blood red in color? Hemoglobin. Exactly. It is made up of what? Hemoglobin. Absolutely right. So now, if everybody accepts, yes, yes. Sanjana and Rakshita, both of you are right. So it is made up of hemoglobin and it is correct. Now, what is hemoglobin? Another question. What is hemoglobin? Hemoglobin is what? It is a protein. Correct? No. What is the role of this protein? Ferrous molecule or the iron molecule. And here you have four chains like this. One chain, two chain, the third chain. What are these chains? These are nothing but peptide chains. You know what is a peptide chain? No, no. So if you see any protein, they have certain structure. The structure of the hemoglobin is nothing but a globulin protein. Why do we call it as a globulin? Every protein can be having a specific. I'm sorry, guys. So, can you hear me? Sir, your voice is not available. Yeah, yeah. Now you can hear me, I think. Just there was suddenly the internet went away. I don't know how suddenly it goes off. Hold on, guys. You can hear me, I think, now, hopefully. If not, please confirm. Yeah. So, thank you. Yeah. So now what I'm saying, proteins are structural molecules. Now we have to study a little bit deeper, and we will study it as we move. Plus one, plus two is also there, and we will be studying again how the proteins are manufactured and how they gain this structure and how we can categorize proteins. Okay. So I think we have studied in the last class like proteins have primary structure, secondary structure, tertiary structure, and quaternary structure. Are you guys being taught this? The proteins can be that they can very sorry, people. Very sorry. Hold on. Might be some technical error. Hold on. Do you answer? You are not audible. Yeah, now I think I'm audible. Chalo. I don't know what is the problem, but suddenly the internet is going even though I have the Wi-Fi. Okay. Okay. Let's see. Let's see. Like we'll continue, and if we have any technical issues, we'll address it later. So now what happens? The structural part of this enzymes is very, very important. Now, how does this enzyme function? So first, what will happen? An enzyme is usually an inactive enzyme. We understood, right? So that they are apoenzymes. So these apoenzymes, an inactive enzyme is made an active enzyme. So what happens? Usually, an enzyme is incomplete and that called as Sorry, people. I think it's raining. That's why there is this problem. Okay. Yeah. So I'm extremely sorry about this. Okay. And I know it's not okay. But anyways, we have to continue. Okay. So this we understood like how an apo enzyme becomes a holo enzyme, and we understood that an inactive enzyme interacts with something called as cofactor. Okay, that is when it becomes a holo enzyme. Now we understand what are cofactors. See guys, cofactors are very important here, and for several reasons. One is it definitely comes in your examinations. Uh, Either it is in the form of examples or it is in the form of uh, is mineral ions a cofactor? So these kind of questions can also be asked. Cofactors can be of three different types. It can be a prosthetic group, it can be coenzymes, or it can be mineral ions. So who is a cofactor? A cofactor can be a prosthetic group. Now I will tell you one thing. A protein, an enzyme, is made up of amino acid. Correct? No, it is a protein. Anything which is non-amino acid, that means it should not be something of amino acid. So these groups are called as a prosthetic group. For example, I told you in the structure of hemoglobin we have iron or heme group. So this iron group is nothing but a prosthetic group. Got it? So similarly, we have peridoxal phosphate. So peridoxal phosphate is also a prosthetic group. So what are prosthetic groups? Prosthetic group is nothing but a type of a cofactor, and these prosthetic groups are non-amino acid components. Okay, they should be of non-amino acids. And example, very easy to understand this example is you have your heme groups. Okay, if you see the structure of hemoglobin, you can see the presence of iron. So this iron, presence of iron is a prosthetic group. Okay, next one. What are coenzymes? Coenzymes, again, this particular part of coenzyme is made up of amino acid component. It is half enzymes, we can tell. You have one half of an enzyme and another half of an enzyme, and these enzymes have to come together, combine to form one functional enzymes or one whole enzyme. So when do we call it as a coenzyme? We call it as a coenzyme when it is made up of amino acid component. What is an example? FAD. What is FAD? Flavin adenine dinucleotide. Okay. FADH, FAD, you'll study as we study photosynthesis. So this is called as coenzymes. Next, you have mineral ions. So mineral ions, they play a very important role. For example, you can see here you have iron for catalase reaction. What is catalase reaction? We have peroxide, H2O2. So when H2O2 reacts with anything, okay, it produces a reaction called as catalysis reaction. So this for this catalytic reactions, what do we need? We need the presence of iron. So when there is iron presence, catalytic reactions will happen. So we tell that it is catalase positive or catalase negative. So for that reaction, what do we need? We need the presence of iron. Similarly, we have salivary amylase in our mouth, and our salivary amylase to function, we need this mineral ion as a cofactor, which is chlorine. So in our mouth we have chlorine, and this chlorine helps us to fill the gap of a cofactor. Did you guys understand this? So it's very easy. Make sure that you understand the three categories of cofactors. It can be prosthetic group, it can be coenzymes, it can be mineral ions, and their examples. Clear? So as I told you, enzymes are highly structural molecules. So now how does the enzymes function? Enzymes function in the form of lock and key model. Similarly, we have a specific lock for a specific key, or a specific key for a specific lock. Sorry, correct, no? So what is our key? Our key is a substrate. What is our lock? Our lock is an enzyme. See, enzyme is here, and substrate comes and attaches to the enzyme at this active sites. Okay, these sites are called as what? Active sites of an enzyme. It cannot attach here, it cannot attach here, or any other sites of attachment. So the site in which a substrate comes and attaches to the enzyme, it is called as active site. Okay, so it comes to the active site and it goes and sits here. So once the substrate comes to the active site of an enzyme and then locks its position, then what will happen, guys? It creates enzyme substrate complex. So now you can ask me a simple question. Now your answer for a complex question is here. So our body undergoes hundreds of biochemical reactions. and we produce hundreds of enzymes. Don't one enzyme react with other enzyme? Don't one substrate react with other substrate? Correct, no? And create a mixed reactions, because this is such a huge chemical factory. Can there be some misfortunes? Can there be some mistakes? There can be mistakes in your lab, but there can never be an error in your body. Why? The enzymes are designed in a very specific way where it attaches to a very specific substrate. That is why all biochemical reactions, even though they are happening all together, they act as an isolated system. You can see their isolated system. They act like an isolated system. Reason being this structural configuration, the structural specificity of an enzyme to its substrate. Okay, you can see here you have an enzyme and the maltose is a substrate with this structure. See how nicely they go and pack. Now there cannot be, most of the time what will happen, there cannot be an identical structure. What happens if there is an identical structure, we'll be discussing further. Understood? So the nature of an enzyme is a lock and key model of enzyme substrate action was actually proposed as a proper scientific discovery. It's not just imagination. Understood? So let's see. One example I will give you here. Now let's say that we have bacteria, guys. Okay? We have bacteria. And this bacteria enters your body. Now our body will kill the bacteria. How does it kill? So one mechanism is you are a soldier. You just attack without knowing the enemy. Simple. And the other one is a very coordinated, very skilled attack, like surgical strike. Very completely, you know who the enemy is. You know how to defeat the enemy, and you have a proper mechanism to defeat the enemy. How does the second thing happen? Usually, our body to kill the bacteria, they produce something. Anything that is coming outside into our body, it produces something called as antibody. Okay. So these antibodies are again proteins. See, not only enzymes, even proteins are highly specific. You understand my point? So this antibody, which is a protein, they are designed in such a way, very specific way, that it will go and attach to the surface of bacteria and recognize, hey, this is a bacterial cell. This is a human cell. So I'm not supposed to touch human cell. I am supposed to kill the bacterial cell. It will be able to differentiate whether it is mine or it is not mine, self or non-self. Similarly, even our enzymatic reactions happen with a very isolated or unique structural configurations. Are you following me, guys? So this is about how the reaction of enzymes takes place. And now we have the concept of activation energy. How do enzymes function, or how do enzymes bring about this efficiency? As I've told you guys, the major requirement of an enzyme is, first thing, we have to make sure that the reaction happens very fast. We need the speed of reaction, first thing. That means it has to consume less time. Within less time, I need more products. That is first, idea. Second thing is, when I'm producing something, I need to make sure that I invest very less energy. Understand my point? So these are the very two important factors that that has to be met to make sure that my system is in profit, or my system is running on good energy. That means if you have more energy to spend, then you will be able to teach or listen to me or run or do other activities. If all my energy is utilized for other things, then you know what will happen to your body. Correct, right? So how does enzyme does this job? Now let us say that we take a chemical reaction, and you see that reaction coordinate. Reaction coordinate means like how the reaction proceeds. Like it's in a positive direction. No, you have two substrate. For example, what is our substrate? We have carbon dioxide, we have water, and then the reaction between these two has to happen. How does the reaction happen? When carbon dioxide reacts with water, it has to give us carbonic acid, okay, H2CO3, okay, it has to give us what? H2CO3. This is our final component. This is our final product. So this is the direction of our chemical reaction. So that is called as reaction coordinate. Understood? Which is a constant. Next one we have is the energy, requirement of energy. Why do we require energy? I require energy because I have to make sure I reorganize, or I have to make bonds, or I have to break bonds before conducting the chemical reaction. In order to do this, in order to combine these two and create bonds, what do I need? I need energy. Correct, no? Now, in a regular case, what will happen? Let us assume that here is where I begin my chemical reaction. Slowly, what will I do? I'll increase my temperature, my pressure, and it goes slowly and slowly and slowly, and it goes to a certain level. So at one point where I have given height, proper temperature and proper pressure, and I've invested so much energy. You see, less energy I invest, less is the product. And once it comes to the optimum energy, so this is called as the highest energy. Okay. So at this point, my rate of reaction is the highest. So now what will happen? The energy required for my reaction, chemical reaction to even take place. So that energy is called as activation energy. Understood? Activation energy is a point at which a chemical reaction starts happening. That means the real combination of carbon dioxide plus water happens at that energy. Understood? So it will take a lot of energy. Now, what will happen with your enzyme? As I have told you, if I have enzyme, okay, first I have what? Substrate. What is my substrate? Carbon dioxide, correct? No? And water. Now, what is my enzyme doing? My enzyme reacts with my carbon dioxide. So this is my enzyme. So now you can see here, this is my substrate. What is my substrate? Carbon dioxide. What is my enzyme? Carbonic anhydrase. Now what are they doing? They create lock key complex. Similarly, you have substrate enzyme. You create a substrate enzyme complex. So here a substrate enzyme complex is created. Why this creation happens? Now, when a substrate and an enzyme reacts with each other, they will try to create alternate routes. You understand my point? So I require less energy to just rearrange the atoms. It's all rearrangement. If I do a small rearrangement, okay, so this rearrangement of the atoms or the molecules, okay, is what is called as the enzyme substrate complex. So when we have an enzyme substrate complex, now for the further reaction, this enzyme substrate reaction will give us what? Product and enzyme. Correct? No, it will give us what? Product and enzyme. So this product and enzyme will be combined form. It will be in the combined form in the beginning. Next, what will happen? The product will split up and then enzymes will split up. So now what happened, guys? We have an enzyme, we have a substrate, they both reacted, they created an enzyme substrate complex. Now, once this complex is created, now the water comes in place. Now they both will react. Who? The enzyme substrate complex and the water will react. Finally, the carbonic acid is created. So once this carbonic acid is created, now this enzyme can no more attach to the substrate. Why it can no more attach to the substrate? Because there is no carbon dioxide. No, carbon dioxide is converted to what? H2CO3. Now the structure has changed. The structure of carbon dioxide has changed. Now once the structure of carbon dioxide has changed, now can it attach to the active site anymore? Definitely not. So this will become as a product and the enzyme will go on its way for the next chemical reaction. So this conversion of enzyme into enzyme substrate complex, now here what will happen? It will take very less energy for this process. So if you compare this, the activation energy without enzyme is very, very high. The activation energy with enzyme is very low or less. Understood? And moreover, you can see that the rate of reaction is also fast. The rate of reaction is also efficient and it is in the positive direction. So are you following me, guys? Next one is, what are the factors that affect the enzymatic reaction? So these are the factors that affect the enzymatic reaction. So it would be temperature, water, pH, substrate concentration. It's very, very important. Next one is the enzyme concentration, presence of inhibitors, and presence of end products. End products in the ones, what is the product that you get? Sometimes as the product concentration increases, the enzyme reaction will also decrease. Understood? So similarly, you have activators or you have radiation, like any gamma radiations or any UV radiations. So they might also reduce the enzymatic reactions. Okay. So these are the factors that generally affect the enzymatic reactions. The most common factors that affect the enzymatic reaction is temperature, pH, and substrate concentration. Remember, guys, the temperature and pH, always we require what? We require something called optimum temperature. AOPTIMUM, yeah. Optimum temperature. What is optimum temperature? at what temperature my chemical reaction is at the highest. So that is called as optimum temperature. You can see here, I keep increasing from 10 to 15 degrees Celsius, 15 to 20 degrees Celsius, 20 to 25. But at all this stage, my enzymatic reaction is not at the highest. It will become highest at the optimum temperature. So this is the temperature at which my chemical reaction can be highest. Now I get greedy. What is my greed? I will try to increase temperature even more. When I increase temperature even more to get more products, now here my enzymatic reactions will not go in a positive direction. It will start deteriorating very, very fastly. Why? Enzymes are proteins and proteins degenerate at high temperature. Okay? Degenerate at high temperature. When temperature increases, as the temperature goes more, my enzyme, what happens? It will become degenerate. All the bonds in the enzymes will break and the enzymes cannot maintain its structural integrity. So when it cannot maintain its structure, it cannot be an active enzyme. So that is why the enzymes will degenerate. When the enzymes will degenerate, I cannot get my product. Similarly, if I go below my optimum temperature, my temperature, I keep on decreasing the temperature. Okay, I keep on decreasing the temperature. Or I have gone below 10 degrees or 0 degree or -2 degree or -5 degree. What will happen to my enzyme? The enzymatic reaction drastically reduces or gradually reduces and it will stop at a point. It will stop at a point. But what happens to the enzyme structure? Enzyme structure is not affected. Enzyme will remain in an inactive state, but it will not degenerate. That is why many of the enzymes are frozen. Okay, you can see that at deep freezing, we can preserve these enzymes so that we can use it for future use. So when you take the frozen enzymes, put it in a normal system. Okay, you take the frozen enzyme and you inject into the human body. What will happen to it? After it gains its temperature to 10, 15, 20, 25, at whatever the temperature, a little bit higher temperature only, it will act. So once that it comes to the optimum temperature, it starts acting again or it starts functioning again. You understood? So in temperature, we have two cases. One is optimum temperature. So as we increase the temperature, at one point, my enzyme reaction or enzyme substrate reaction is at the highest. So this is called as what? Optimum temperature. So below this or above this, I cannot get the same, quantity or same production I cannot get. So this temperature is called as what? Optimum temperature. Now, what happens to the enzymes when I increase the temperature? As I keep on increasing the temperature, my enzyme activity will suddenly decrease or very fastly it will decrease. And eventually, my enzymes will degenerate. Why enzymes will degenerate? Because enzymes are proteins and proteins are very highly sensitive to temperature. They're highly sensitive to temperature. So when you go beyond 45 degrees Celsius, usually most of the enzymes will start degenerating and they will not function. Gone. Those enzymes are gone, destroyed. Now, this is first another case. Now, if I decrease the temperature, if I keep decreasing the temperature, what will happen to the enzymatic activity? The enzymatic activity will keep on reducing and it will stop. Now, what happens to the enzyme? The enzyme will remain inactive, but it will not degenerate. Did you guys understand the temperature as a factor affecting the enzymatic reaction? Yeah. Very important concept, guys. Like biomolecules is a very important chapter because a lot of questions can come here. Okay. 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 Inactive enzymes by decreasing temperature. Inactive, yeah, inactive